It's my great pleasure to introduce our first speaker, Bruno, who I have known since he was a really, really young graduate student. Before Bruno joined Greenberg's lab in Harvard this year, he was a graduate student in Cold Spring Harbor lab, where I have met him. He was performing his PhD thesis with Jessica Token. He's probably one of the most hardworking person in Cold Spring Harbor campus, works much harder than I do. And he not only works really hard, but also have thought really critically about his project and really learned a lot of new techniques, and also shared all his expertise, especially the low cell number epigenomic assays with the community as well as outside the community, including serving as instructors in different courses for cut and run. He has really taken advantage of these newly developed methodologies, such as cut and run and single-cell RNA-seq, to look into how estrogen receptors regulate different neuron populations, and published a really wonderful story in Nature earlier this year, which is a really well-deserved publication. And then his study has shed lights on this long-standing question in terms of where estrogen receptor binds in different neural populations. And I will give him the floor to tell us more about this awesome story. So with that, I would hand the stage to Bruno.
Okay, awesome. Thank you, Sky, for the great introduction, and thanks for the invitation to talk today and talk about my work. Okay. So yes, as Sky mentioned, today I'm going to be talking mostly about my graduate work studying how sex differences in their brain at the level are regulated at the level of the epigenome and gene regulation. So I'm a neuroscientist, and I'm very strongly interested in this central question of how the genome specifies these innate behaviors that animals throughout the animal kingdom display, behaviors that typically require no training and are expressed innately. And so to sort of work backwards from what we can observe in the laboratory when we just look at, lab mice, a lot of these behaviors are controlled by discrete neural circuits in the brain, which are comprised of individual neurons. And so I'm very interested in looking at how the genome, and specifically how gene expression is regulated, gives rise to innate behaviors by acting within these discrete neural populations in the brain. And it turns out that sex differences actually provide quite a good model system to understand this fundamental question of how the genome programs innate behaviors. And the reason for this is because all animals essentially display sex differences in these critical innate behaviors that are required for survival and reproduction. And these range from aggression behavior, mating behavior, parenting, and many more. And it's been known for a little over 60 years now that sex differences in behavior are largely regulated by the release of gonadal hormones. And so there was a landmark study back in 1959, which established this fundamental hypothesis in the field called the organizational activational hypothesis. And this hypothesis basically states that gonadal hormones act during different times in life to regulate sex differences in behavior. So early life production of gonadal hormones actually act to organize the underlying neural circuits that control sex-typical behavior. Subsequently, after puberty, there's the adult production of gonadal hormones that then act upon these same neural circuits to activate sex-typical behaviors. And so this is kind of the schematic of when these hormones are being produced in mice, which are the model system that I use to study sex differences in the brain behavior, in relation to sort of key neurodevelopmental events that are going on throughout the brain. And so male mice actually have a surge of testosterone right on the day of birth that coincides with major developmental events such as apoptosis and synaptogenesis. Then there's a period of quiescence, and then puberty happens, after which mice produce testosterone. The males do, or females produce estradiol via the ovaries. Surprising finding from a little over 10 years ago was that it's actually not testosterone itself that's critical for specifying sex differences in the brain, but it's actually the conversion of testosterone into estradiol locally by the aromatase enzyme that's important for organizing sex differences in the brain. In adulthood, testosterone production in the males activates these male-typical behaviors such as mounting, aggression, and territory marking, whereas estradiol in females activates sexual receptivity and parental care. And then I'm just showing here in parallel what these hormone surges look like in humans, just to show you that this sort of overall logic of when hormones are being produced is relatively conserved across species. Given that these hormones are important for regulating behavior, it should come as no surprise that the receptors for these hormones are genetically required for the display of sex-typical behaviors. So this is data going back over 20 years ago now, since near the beginning of mouse genetics, in which Donald Pfaff's group actually generated constitutive ER-alpha knockout animals, or estrogen receptor knockout animals. And they found that if you remove the receptor for estrogen, you can prevent the display of female typical sexual receptivity behavior, which is called lordosis. Likewise, if you remove estrogen receptor in the males, you can basically block that early life organization and prevent the display of male typical aggression, both sort of the frequency of the aggressions, so how many times a male will attack another male, and also the intensity of aggressive behavior in terms of the duration. And so the question then is, what are hormone receptors and what are they doing to then mediate sex typical behaviors in the brain? And so hormone receptors are essentially transcription factors. The primary hormone receptors are estrogen receptor alpha, or ER-alpha, androgen receptor, which is the receptor for testosterone. There's also progesterone receptor, mineralocorticoid receptor, and glucocorticoid receptor as well. And these TFs are ligand-dependent transcription factors, so they bind to their hormone, and then upon binding, they actually dimerize and then bind to their recognition sequences throughout the genome. And hormone receptors are pretty ubiquitously expressed throughout the body, where they regulate many different biological processes outside the brain. And so this is just a model figure showing where ER-alpha is expressed, all the different tissues in which it's expressed, and all the different biological functions it's known to regulate. In the brain, ER-alpha is expressed within sparse, discrete neuronal populations, primarily in the hypothalamus and the amygdala. And its underlying genomic mechanism and cellular mechanisms remain largely unknown. And so this is sort of the starting point for my graduate work, which is getting at this fundamental question of how do hormone receptors generate sex differences in the brain and behavior. And the underlying hypothesis, given that we know that they're transcription factors, is that really it's gene regulation by these receptors, which enhancers they're binding to and which genes are actually controlling, is how the genome specifies the display of innate social behaviors. And so my work has largely looked within the specific populations of neurons in the brain that are important for regulating these sex-typical behaviors, and then applying a number of different genomic assays and transcriptomic assays to really identify where the receptors are binding in the genome and which genes they're controlling. I've just listed several of these techniques here. I'm going to be talking mostly about cut and run and single-cell multi-omic data. And I should preface this by saying that the targets of these gonadal sex hormones in the brain are largely unknown prior to this study. And the reason for this is because of the technical limitations of ChIP-seq. So typical ChIP-seq experiments for transcription factors often require, at least 1 million cells, depending on how abundantly expressed your receptor is or your factor is. And so for this reason, ER alpha function has mostly been studied in human breast cancer cell lines and breast tumor biopsies, as well as a number of peripheral mouse tissues where it's abundantly expressed. So for this project, we benefited a lot from the development of cut and run, which is sort of a low input transcription factor profiling method developed in Steve Henikoff's lab back in 2017. And so the first thing that I did was basically take these key brain regions in the hypothalamus that are important for the display of sex-typical behaviors, and then basically treated animals with estradiol or vehicle control, and then isolated nuclei from these regions for ER alpha cut and run. And after some optimization, I was able to identify around 2,000 sites in the genome where ER alpha is recruited to in response to ligand. And these sites are primarily located at distal enhancers, either in energenic regions or also intronic regions as well, as opposed to promoter binding sites. And the majority of these sites actually happen to be specific to the brain when we compared it to different mouse tissues. And this is just an example of a binding event where we can see ER alpha targeting neurexin gene, which is a gene important for synaptic cell adhesion, specifically in the brain, as opposed to a binding site for ER alpha that's actually ubiquitously shared across many different tissues. And so once we got this technology working, I was really interested in getting back at this fundamental question of how the hormone surges, how the neonatal hormone surge actually organizes sex differences in brain development. And so there's two kind of major schools of thought for this field. The first of which is that sex differences are purely observed at the circuit level. And this is supported by many solid studies over the past few years, which have really looked at axonogenesis and cell survival in response to the neonatal hormone surge. But there's also this other idea that sex differences might be regulated at the level of the epigenome, and that the consequences of early life hormone signaling might persist at the level of gene regulation. And this is really supported by in vitro knowledge from what we know that the cofactors of hormone receptors in breast cancer cell lines. But we actually know very little about where ER alpha is binding to the genome and which genes it's regulating following this early life event. And so basically, I optimized an approach to look specifically within the BNST, which is a key node in the circuitry of sex-typical behaviors, to identify sex differences in the chromatin landscape following the early life hormone surge. And I utilized this genetic mouse line that's called SUN1 GFP, which basically expresses this GFP nuclear reporter in a Cre-dependent manner. So we can cross it. I crossed it with our ESR1 Cre mouse line, ESR1 being the gene for ER alpha, so we can selectively pull out the ER alpha expressing nuclei from this brain region. And so looking 4 days after this neonatal hormone surge, we can identify around 2,000 sites in the genome that actually differ between females and males, with the majority of these sites actually opening up as after this hormone surge occurs. To determine whether these are actually dependent on the neonatal hormone production, in parallel, I actually treated females at birth with estradiol. And I find that females treated with estradiol, which has previously been shown to masculinize behavior, similarly masculinizes the chromatin landscape of these ER alpha expressing nuclei in the developing brain. Furthermore, to figure out if ER alpha is actually causing the chromatin to open up, I performed cut and run on the day of birth, selectively in these ER alpha neurons for ER alpha itself. And I can find that ER alpha is being rapidly recruited to these sites that are opening up as a result of the hormone surge, which really demonstrates that ER alpha is being engaged by this early life hormone event to specify sex differences in the chromatin landscape. And so these experiments were all done in bulk populations of ER alpha neurons in this brain region. But we know now from many different studies over the past few years that almost all regions in the brain are comprised of heterogeneous populations of neurons. And so I really wanted to look at the level of specific neuron types to see how sex differences are regulated. And to do that, I optimized and applied this recent single nucleus multiome sequencing approach from 10x Genomics, where you can actually capture the RNA and attack from the same individual cell. And I applied this to 2 different developmental time points following the hormone surge, postnatal day 4 and postnatal day 14, to identify whether sex differences are actually persisting long after this hormone surge completes. And so this is the UMAP of the cell types that we identified in our brain region. I mapped this to an adult reference that we had previously collected from this the same brain region. And we can identify that most of these cell types are actually strongly conserved across developmental time and across sexes. What was most interesting is that we can actually detect sex differences in gene expression that vary a lot across neuron types within this region. So certain populations of neurons have additional or higher numbers of sex differences, where others have very few, despite the fact that they all express E or alpha. And moreover, when we look actually at the chromatin landscape, because this is multi-omic, when we also have the attack data, we can see that this previously defined list of sites that are opening up at the bulk level are actually pretty heterogeneously regulated across neuron types. So each row here corresponds to a different neuron type in our data set. And basically the signal here is the difference between males and females at individual sites. So around half of these sites are really strongly sex biased within a specific neuron type, whereas the other half seems to be regulated across multiple neuron types. And this is just an example of what that heat map is representing. So you might have, this is 2 specific genes that I just pulled off of this list. One is a serotonin receptor, another is important for ECM remodeling. And we can now detect that these genes are actually sex biased within particular cell types in our data set, and actually map the specific enhancers that are critical for regulating the sex biased expression of those genes. So this is an example where the serotonin receptor is male biased, specifically in this I1 inhibitory neuron population. And this is an example of a male bias enhancer that is open in this I3 population. And so this idea is that we can now use this information to identify what these specific neuron types are doing during brain sexual differentiation. What was really interesting about this is that we can actually see that some sex differences persist across this neonatal critical window. So these are 2 example genes and enhancers for which both the gene and the enhancer persist the sex bias between P4 and P14. So this is long after the neonatal hormone production subsides. And we see many more examples of these types of genes. They don't seem to be random, but they seem to be particularly relevant for the neurodevelopmental events happening at that time, such as axon outgrowth and synapse formation. And so we think that this persistency at the epigenomic level may coordinate sexual differentiation over this prolonged 2-week critical period. And then just finally, I'd like to mention that we did experiments to look to see whether or not ER alpha is actually required genetically for sex differences at the behavioral level, and also at the level of gene regulation. And so what I didn't mention at the beginning of my talk is that we had previously found that if you knock out ER alpha, either in excitatory neurons or inhibitory neurons in the brain, you get different phenotypic effects. So knocking ER alpha out in excitatory neurons has no effect on sex typical behaviors. But if you remove ER alpha in inhibitory neurons, you can actually impair male typical aggression and territory marking behavior. And that's part of the reason why we know that certain populations of neurons in the brain are more important for being organized following this neonatal hormone surge. So inhibitory neuron deletion of ER alpha primarily affects the BNST the medial amygdala, and the medial preoptic area. So these are important brain regions in the hypothalamus and amygdala that are important for regulating sex-typical behavior. Likewise, what I did was basically look to see if you remove ER alpha, do you ablate this transcriptional program that I had defined in our multi-omic data? And so I basically generated a single-cell RNA-seq data set of ER alpha knockouts in inhibitory neurons from male animals, as well as female and male control animals. And similar like before, mapped these to our adult reference atlas, and then was able to identify that if you remove ER alpha conditionally in the males, you can actually prevent sex differences in gene expression from occurring, really demonstrating that this transcriptional program is likely subserving the sex differences that are observed in behavior later on in adult life. So in summary for this talk, when I first started my PhD, I optimized several of these approaches for looking at the binding of gonadal hormone receptors in the brain. And I think the next exciting thing will be to expand this to look at other hormone receptors to figure out what they're doing, such as androgen receptor and progesterone receptor. And secondly, once we'd established this approach, I basically applied this to early brain development to demonstrate that this neonatal activation of ER alpha is really driving a sustained gene regulatory program that's critical for sexual differentiation of the brain and behavior. And that's schematized over in this panel on the right here. So sort of major open questions for the future. This was pretty much all done with transcription factor binding and ATAC-seq, so mapping the chromatin accessibility landscape. We still know actually very little about how hormone receptors are influencing the epigenomic landscape of both developing and adult neurons. We don't know, for instance, whether it's regulating DNA methylation, various histone post-translational modifications, aspects of the 3D genome. There's a lot known about this in breast cancer, but very little known about what it's doing in the brain. And this might also shed some light on which cofactors ER alpha might be recruiting to then mediate these effects. Second major open question is not only at the genomic level, but now looking at the cellular level to really understand how the downstream targets of ER alpha give rise to sex differences in BNST circuitry. So now that we've mapped these targets, the important question will be then to understand how does this gene act within this particular cell type to give rise to a sex difference in axon outgrowth or cell survival. And so with that, I'd just like to thank my members of my lab who helped support this research over the past few years, particularly Jessica, my former PI, and Melody, our lab manager, who helped a lot with some in situ data and also generating knockout animals. The various core facilities at Cold Spring Harbor that help support this research, my funding, and thank you all for listening today.
Thanks, Bruno, for sharing his PhD work. And if you have any questions, please feel free to type it in the Q&A box, and Bruno will answer them during the rest of the seminar. And I just have a very quick question. So you mentioned all these different receptors. Do they influence each other's binding, or you think it's just one dominating?
Yeah, so I do think that they do. There's certainly a high degree of co-expression of ER alpha with androgen receptor and also with progesterone receptor. And there's some great work from Jason Carroll's lab, who has really done a lot to understand what ER alpha is doing at the genomic level in breast cancer, demonstrating that ER alpha can actually influence AR recruitment and also progesterone receptor recruitment. So an interesting hypothesis in this system, since we also know that AR is important for activating sex differences in adulthood, is that ER alpha might somehow influence the binding of AR later on in adult life. And I think our lab is doing some experiments now to look at that hypothesis.
Yeah, that's really cool. Yeah, thank you for your seminar on the